Imagine a cube of ice generated by your freezer at home. The white coloring is due to air bubbles trapped and frozen in the water ice. As the planet cools, the same mechanism allows for bubbles to appear beneath the surface of the crust, trapped in place by the frozen lava. Some of these bubbles became geothermal hot springs, where vents reaching into the superheated ocean pumped primordial soup into the rocky cavities. Alexander Oparin, a Russian biochemist working independently alongside a British scientist, J.B.S. Haldane, published a book in 1924 which he titled The Origin of Life. It described a speculative process where life could have originated from. Unfortunately, at the time nobody could come up with an experiment to showcase such a process. That is until about 3 decades later. The Miller-Urey experiment, performed in 1953 by Stanley Miller and Harold Urey, was an attempt to create a simulation of the atmosphere of early Earth before the advent of prokaryotic life. They used water, methane, ammonia, and hydrogen gas in a tube to simulate the contents of the atmosphere. Then light, heat, and arcs of electricity are zapped through the precursors where the effects are then observed. In 2007, after the death of Miller, the vials of the original experiment were examined and were found to contain well over 20 different amino acids produced from the original experiment. Organic molecules were proven to be able to be created from parts of inorganic molecules stirred by the hand of heat and radiation. Around this time, the atmosphere in Dianthos is still highly pressurized, but slowly decreases in this stage from about 200 atmospheres of pressure down to a stable 2 Earth atmospheres, or 15-20 millimeters of mercury. The air is made up of 21% carbon dioxide, 5.3% hydrogen, 1.62% argon, with the remaining 72.08% composed of nitrogen. Deep in geothermal hot springs and along volcanic fault lines, the hot gas and nutrient-rich biome cause reactions to start happening spontaneously, forming amino acids and simple sugars. These processes can create soluble and insoluble products that over time alter the environment they occur in. The first life was not even a cell in Dianthos, just a pattern of chemical reactions. After all, reaction C can't occur without substance B in the environment, which all starts from compounds produced by reaction A. This then allows for the existence of other reactions to espouse from the fresh new products. Some of the byproducts from one reaction feed into another reaction. Therefore, the first life did not even exist in a cell and occurred piecewise over eons. To prevent taking on the burden of regulating the entire ocean, systems developed a shield to seal themselves off from the rest of the world. However, one type of early life did not take this route. The most simple type of early life came about in deep water caves. These are the systems that never developed cell walls, the informe domain, Latin for shapeless. Informe take up the volume of their container and work to maintain homeostasis around a source of fresh material. Since there is no life yet on this planet, there is no competition to their survival save for acts of God. The caves which give Informe their properties are always sealed on the bottom as to allow sediment to deposit, with a geothermal vent underneath. They also sometimes had nearby pockets of air, allowing for exchange of gas with water. These caves are aptly named dens or stomachs due to their highly acidic environment. In open areas of sea that are exposed to the air and ultraviolet radiation of a sun twice as luminous to that of Earth's is where true life begins to develop. This takes a lot longer than the cave habitating Informe took to develop, as the environment is much less predictable on the open seas. Some theorize that leaks of Informe dens into the waters cause cellular life to develop, but that is still being researched. The complexity of life is measured by the emergent properties of a living thing that come from simpler components. For example, beings with tissues are more complex than cells. Cells make up tissues which make up organs. Objects that use an automatic system of organs are creatures or animals, even plants. This trend could continue, as a system of beings could be an economy. Economies working in tandem start to make us seem like uncoordinated little ants. On planet Dianthos, the first systems were a lower level than that of cells. Above Informe, in the stages of complexity, cells can be found. Unicellular organisms were the first to conduct the chemical operations within the confines of a wall or membrane. Cells require 3 aspects to be considered true cells: membrane production or repair, energy generation, and duplication of DNA and RNA. This satisfies the rules of life: reproduction, metabolism, development, response, and homeostasis. Each enzyme usually only facilitates 1 or 2 functions. Motor proteins carry substances from one end of the cell to the other by crawling along microtubules of the cytoskeleton. The protein is aware only of its purpose and does not have any awareness of its immediate local environment. However, groups of proteins can collectively sense a concentration of specific target molecules. So the cell acts like a machine, akin to a mailing center sending out envelopes that arrive to their area of work. For cellular life to exist, it will be dependent on its environment for a source of nourishment. The first cells won't have any kind of mobility, nor can they breathe or even process any gas, making them lazy little heterotrophs. Eventually, there will be hundreds of different species of these guys all over the planet, recycling usable carbon for their descendants to harvest. The very first cellular life did not have a full separation between inside and outside. It was made purely of a combination of hydrophobic amino acids with intersects of enzymatic proteins and amino acid production. Like a sponge, water flowed through the holes in its barrier to transport compounds. The barrier did act like a sieve and filtered out large harmful molecules. These will be known as falsi ipsia. The name of the other root category of simple heterotrophic prokaryotes are virusia ipsia, or true selves. Their membrane demarcates the boundary between a self and an external environment. V ipsia membranes are composed of a mix of phospholipids and proteins. From now on, ipsia refers to members of virusia ipsia, singular being ipsum. Because phospholipids naturally form into spheres, called micelles, it's only natural for a cell to use this for its own purposes. The head of a phospholipid loves water. It's hydrophilic, and the tail shies away, hydrophobic, causing a sphere to form, a micelle, where tails are pointed inward and the head is outward. A large enough micelle has phospholipids fall to the center, forming a space inside the body. This is called a liposome and is easily recognizable as a blueprint of a cell. While chemical pathways allow for the synthesis of phospholipids, emulsifiers regulate the amount created with fine precision, as any excess fatty acids are connected on the nonpolar end, while polar water attaches on the other. This can allow for disposal of excess lipids easily. An example of an emulsifier in your life, hopefully, is soap, which helps water to wash away greasy lipids. Energy generation on planet Dianthos occurred in stages of ever-increasing complexity. Informe are classified as chemotrophs, as they harness energy from combining inorganic molecules in their dens with oxygen in the air and rocks. The first life on Dianthos was autotrophic. There are many metabolic pathways for bound sulfur in the environment to be converted into H2S. One form of Informe stomach was what is known as a kip generator, using the sulfur emitted by vents at the bottom of their dens in order to convert sulfur, formaldehyde, and water into carbon dioxide and hydrogen sulfide. Hydrogen sulfide is water soluble, which causes pH levels in the stomach to drop. Another uses iron to metabolize it. In order for this to work, there has to be plenty of iron in the environment. Thankfully, iron is quite abundant on prehistoric Dianthos. One metabolic pathway for the Informe to produce hydrogen sulfide is this. Anyways, all pathways utilized by the Informe are non-enzymatic. The wide availability of sulfur and hydrogen sulfide in the environment, thanks to volcanic activity and the hard-working Informe, gave the passive Ipsia a vital source of energy. At first, Ipsia developed metabolic pathways, a kind of microorganism digestive system. It synthesizes ATP from compounds in the environment. Newly developed enzymes can create and interact with hydrogen sulfide, using its properties to combine carbon dioxide and water into glucose. This special guy will be a surprise mouse tool that can help us later. In order for one cell to recreate itself, it must be able to pass on instructions of how it's structured and how to replicate itself again. The grittiest of details will have to be glossed over for the purposes of the length of this video and my sanity. This is an alien evolution project after all. However, it is easy to imagine that a population of elementary reaction chains could leave behind its own building blocks as it lyses. GNA is the result of some of the first macromolecules, molecules that can be hundreds to thousands of atoms big, experimenting with hydrogen bonds. It is possibly the simplest nucleic acid. However, it gets outcompeted by threose, a precursor to RNA. RNA, when it is invented, becomes a driving force to pass the torch of life. This molecule could bond to amino acids, creating ribosome-like structures. The activity of these nucleoproteins will eventually give way to a much more efficient DNA-protein dichotomy. If the instructions could hold on to itself, it becomes a much stronger structure than a loose string. Therefore, the earliest of animalcules tied their proto-RNA in loops. This advent of technology allowed for a much longer lifespans, in the range of dozens of minutes. In the sulfuric seas, these Ouroborea ballooned in population over the coasts of the continent. Over time, they will evolve into living membranous toroids. DNA, a double helical structure, is more protected and more advanced than RNA. Fast forwarding in time, we can see the evolution of this molecule. This project will use classic DNA molecules to do this with very minor variation. Early cellular life may rely on manipulating compounds similar to ATP, such as GTP, CTP, UTP, and TTP. However, these vary slightly from one to the next and have their own unique use cases. These will be ironed out by the invisible hand of evolution over passing time. Protection of the DNA is achieved by the development of a reinforced inner vacuole. The pliable inner layer of phospholipids is separated from the outer layer, while being filled back in with quickly constructed hydrophobic amino acids that are slowly replaced with newly synthesized, more complex phospholipids. Because the nucleus is the first structure to form inside the cell after its birth, the most important structures created soon after will use it as an anchor point to build off of. This is where a phospholipid factory is most likely to develop. Some branches of V. ipsi have this factory built at the same time as the nucleus by splitting the diverging membrane layer into a double-headed shape. It requires a more complex system to prevent harm to the membranes as it is separated. This causes a double nucleus effect. In both types of cell, the phospholipid factory is known as the orogenoplasmic apparatus, meaning the from-membrane structure. Duplication of DNA is done in a manner similar to Earth, where the three main stages are transcription, translation, and duplication. The exact chemicals and enzymes were made up over generations of botched attempts and in no way reflect Earth's. However, as the main steps are the same, the effect is identical, such that DNA is copied over with limited mistakes. In most cases. Once untied, one half of the DNA strand is matched and shot out one side of the duplicator complex. The other is read backwards and must be looped around every so often after a certain number of nucleotides are read in order to sync it back up with the next part of the data chain. Since the straight DNA is copied faster than the backwards DNA, that backwards one is sent to where the daughter cell will erupt since it has farther to go. This synchronization allows for quick splitting of the cell into two daughter cells. During mitosis, the cytoskeleton is no longer useful and is broken up into many parts and recycled by lysotubulase. Floating parts are then reused to create the spindle fibers that are sewn through special ports of the chromatin blocks. Once securely attached, the spindle fibers are coated in a layer of kinetochrome, which acts like a train track for motor proteins. The combined action of the fibers and its new layer act like a typical kinetochore on Earth. They swarm the fibers and get to work pulling the DNA into two sections. An abundance of usable organic matter leads our populations to advance and specialize into many clades. The next generation of squirmies is laid out by a combination in differences in energy production and code distribution. Some of these cells have developed pigments. Photosynthetic metabolism emerges from the harnessing of the ultraviolet radiation of the sun. This process evolved from a mutation of some orogenoplasmic apparati, or what was originally produced as a byproduct, made its way into the facility corridors and built up as plaque does on your teeth. Worker proteins in the cell first collect them into a mass for expulsion. Later down the evolutionary line, they are used to direct sunlight into H2O bonds to release the energy inside in a process known as photolysis. Pigments come in a variety of shades. Melanins are made from the process of oxidizing tyrosine catalyzed by tyrosinase and come in red, brown, and black colors. Indigoids are often overshadowed by darker tones, but are lighter shades of purple, blue, red, and green. The sun's color temperature at this time measured at 69,420 Kelvin, 6541 Kelvin, making it appear almost white. Therefore, the most popular rudimentary pigment is black, as it encapsulates an inefficient range of colored light on the spectrum. However, sulfate-reducing bacteria turn a purple color due to indigo pigmentation. Melanocytes are organisms that developed a black pigment. They collect near the surface of the water in large flat groups. Others later turn to anaerobic phosphorylation, a process by which a cell can expend 2 ATP to generate 4 ATP by transforming glucose into lactate. This is a very inefficient means of energy production. Glucose can yield up to 38 ATP, but it is far superior to no energy production at all. Yet still more bacteria sink to the lower parts of the ocean, where sunlight is weak and cannot permeate. The concentrations of glucose can reach them where they can metabolize it, producing methane as a waste. Cells that developed photosynthesis have no reason to focus on mobility, instead choosing to reinforce their shells against the shifting tide and sulfuric environment. Photosynthetic Pyridus thoracosminia start using silicates in their membrane, which allows for some beautiful shapes. These are like the diatoms of Earth. Where the eating was good in the shallow waters, some thoracosminia evolved hook-shaped corners to their shells to stack together. This caused piles of organic matter to form and solidify over the decades in a manner similar to that of stromatolites on Earth. These are the Hackenphytia. Bacteria in thoracosminia grew up on a planet with certain atmospheric conditions. However, because photosynthesis involves the expelling of gas as a byproduct, and was such an efficient and useful energy system, this gas exploded into the atmosphere. At this point on Physeden, high levels of greenhouse gases like carbon dioxide and methane are vibing in the atmosphere. Oxygen combines with the methane to form carbon dioxide and water, while at the same time, bacteria in the water are sucking carbon dioxide from the atmosphere, stealing the carbon to feast on, and releasing the oxygen back into the air. The once reducing atmosphere of Dianthos became an oxidizing one after the rise of biologically produced dioxygen. The gas settles in the stratosphere as the buoyancy of the gas guides its altitude. High-frequency rays that normally would contribute to heating the surface of the planet are instead petered out miles above the rocky surface, their energy being used to break up dioxygen molecules into lone oxygen atoms, which quickly latch onto other dioxygen molecules, forming molecules of ozone. Because life developed in an environment lacking in oxygen, she was not prepared to deal with the effects that come with an environment that inhibits reactions that are helped along by reducing atmospheres. That much oxygen is plainly unnatural and caused a large die-off of microorganic life that could not survive in the toxic oxygenated environment. Oxidizing atmospheres contribute to very different reactions such as combustion and rust. In fact, much of the ferrous iron, iron 2, was converted to iron 3. Ferric oxide and ferric chloride became abundant in the sediments of the water. Dissolved iron in the water was turned into magnetite. What may be surprising is that it wasn't necessarily the abundance of oxygen that led to complex eukaryotic and multicellular life, but the levels of oxidized iron. After all, almost all known forms of life, particularly complex life, require iron. The reason iron is necessary for complex life is due to the creation of the relatively simple ferritin that all complex beings are required to produce. Beings with ferritin are classified as ferrophyllum. With the advent of ferrophyllum, let's go over the entire phylogenetic tree of our single-cellular life on Dianthos. Here we see a map of the first period of life on the planet. Starting at the top, the root of Informe splits off into three categories: the Falsia ipsia, Verusia ipsia, and the Siderophagia. The Siderophagia, meaning iron-eating, become the future Siderechon, meaning iron-bodied. The Falsia ipsia, on the other hand, turns into the Autohymenia, the self-replicating walls. However, the real party begins with the Verusia ipsia, the true selves, splitting off into the Ferrophilia and the Ouroborea. Following the Ouroboros tree from left to right, we have the external organa, the outside organs. The Latanza, meaning the wide loop, splits into the tubulotextare, the tube weave, and the tubulomosia, from there, the tube-shaped. The Latanza also turns into the retortanza, the twisted loop, which become the lameotansia and the cochleophylutansia. The retortanza also splits into the flagellotanza. Finally, from the Ouroborea, we get the denihemicosmia, the thick half-loops. Going back to the Ferrophilia, we get the homeosporia, which have 4 nucleotides. These have DNA and contain oraginoplasmic apparatuses. They become the ensepkaryotes, the double-headed kernels, which split into the duodenostemma, with 2 stems, and the duplicofronzia. Homeosporia also gives us the euhomeosporia, which contain the teophagia, purple sulfur-eating bacteria, and the methanopsia, the lactate producers and methane producers. Here, potentially the most important clade of Homeosporia, we get the Pygmatosporia, which contains the inanoplastids, the empty plastids, and the ripanoplastids, the ones that store protein, fat, and starch. We also get the chromoplastids, which split into the indigoplastids, melanoplastids, which become Hackenphytia from before, the chromatoplastids, which contain red, orange, and yellow hues for the rhodo, auranti, and citrinoplastids. And finally, the viridioplastids, which contain pyritidia, euviridia, fibropharia, and chelacoformia. Fibropharia meaning fiber-shaped, and chelacoformia meaning match-shaped. This wraps up the Tree of Life for the first stage on Dianthos. Thank you for watching. Hey, and for anyone that made it all the way through, please drop a subscribe. I'm only going to be making good content, as you can see. Since you made it this far, you can tell. And I'd like to shout out some of the people that have made this possible for me personally to complete this part of the project as an inspiration. They mostly have YouTube channels, so I'm going to go from most subscribers to least subscribers. So here we go. I want to thank Spore, who's inspired thousands of people in their own projects. Curious Archive makes some great videos. We got Mothlight Media. I like that channel a lot. Thank you for making those. Artifexian, world building right there. Bibloradion, that guy was a lot of help getting me started. And now we get to some of the unknown ones. Project Rose, that guy has some really cool videos. Nano Rooms, you must check those out. Those are just great. And personally, Candy Eggs. This guy has a lot of weird videos, but his number 1 and number 2, especially the second one, is just top quality project right there. There's a curveball right here. Michael Levin graduated from Tufts University, got his PhD from Harvard, and he currently teaches at Tufts University. Probably one of the most influential minds of this generation. And finally, Liam Danger Park. He's an ADHD coach, and he inspired me to actually put my mind to this project and work out all the gritty details and things, learning Adobe After Effects and Blender. You haven't seen that yet, but it's coming. Man, I really hope I'm not forgetting anything. Man, I could have sworn there was one more that's just on the tip of my tongue, and I don't remember. Oh well. You have a great day. Bye-bye.